So I'm going to start a new series on high yield topics from the biochemistry. So let's just discuss elastin and collagen today. So firstly, let's discuss about collagen. Collagen provides rigidity to the tissue structure, whereas the elastin provides elasticity to any tissue structure. So firstly, discuss about collagen. Uh, the synthesis of collagen occurs in the rough endoplasmic reticulum, in which there is firstly from the mRNA from the mRNA there will be synthesis of this pre-pro alpha chain into the RER, and in case of the pre-pro alpha chain, there will be a presence of a hydrophobic sequence which is attached to the pro alpha chain of the collagen, and after the action of peptidases, this hydrophobic sequence is removed, which will result in the formation of the pro alpha chain. So there are a lot of a lot of lysine and proline residues which is attached to the pro alpha chain, and this lysine and proline residues are now hydroxylated in the presence of vitamin C as a cofactor. So hydroxylation of lysine and proline residues occurs, resulting in the formation of pro alpha chain. In the first step, all the hydroxylysine residues are glycosylated, not the hydroxyproline, but hydroxylysine are only glycosylated, resulting in the formation of a final pro alpha chain structure. So this pro alpha chain structure now undergoes coupling. So three alpha chains, three pro alpha chains, which has gone the process of glycosylation, undergoes trihelix formation to form a molecule called procollagen. So these three pro alpha chain will form a trihelix to form procollagen. So now the N terminus and the C terminus of the procollagen has to be removed. For that, it has to go out of the rough endoplasmic reticulum into the cytosol and then into the extracellular matrix, in which our procollagen structure gets cleaved at the N terminus and C terminus by the action of peptidases. So our final collagen structure is formed, and this is also called the tropocollagen, in which the N and C terminus have been cleaved. So now cross-linking of this collagen molecule occurs between other collagen molecules in the action of lysyl oxidase, which uses copper as its cofactor. The different collagen molecules are now cross-linked to form a collagen fibril, which is a fibril structure, and finally it undergoes further aggregation to form a collagen fiber. So this is the synthesis of the collagen. So let us firstly understand that for the formation of the trihelix, the collagen molecule is always has a structure, trihelix structure, tripeptide structure of glycine X and Y, and the X and Y may be any kind of amino acid residues, because the glycine is a very small amino acid and it helps in a proper trihelix formation. So the side chains of the glycine is very short, so it helps in proper trihelix formation. So this all process is happening in the fibroblast, inside the RER of the fibroblast, inside the RER of the fibroblast. So let us discuss about some high-yield diseases on collagen. So firstly, we have osteogenesis imperfecta. This is a type 1 collagen defect. Occurs as a defect in COL-A1 and COL-A2 gene, and the main defect lies in the trihelix formation and is characterized by blue sclera, hearing loss, and multiple fractures. The next disease would be scurvy. In scurvy, we have our vitamin C deficiency. So as a result, the hydroxylation of the proline and lysine residues does not occur, and as a result, it is characterized by bleeding gums. Similarly, another disease we have Ehlers-Danlos syndrome. So in case of Ehlers-Danlos, this lysyl oxidase enzyme is in defect, or another enzyme which is our peptidase, which cleaves the enzyme C terminus, is also defective. So as a result, our final collagen fiber is not formed. So our collagen fiber is necessary to maintain the rigidity of the tissues, which is lost. So as a result, the skin becomes hyperlaxicity. There will be hyperelastic skin, hypermobile joints, and easy bruising of the tissues, which is often categorized as classical type. In case of vascular type, we have CNS aneurysm, where blood vessels are not elastic enough and undergoes aneurysm. And another disease is the Alport, which is similar to the osteogenesis imperfecta. So there is collagen 4 defect. And in case of Menkes disease, we classified Menkes disease as kinky hair syndrome. So in case of Menkes disease, the child comes into the OPD with a growth retardation, and upon arteriogram there will be tortuosity of the major arteries, and there will also be hematomas, like subdural hematomas in the child. Also there will also be other features like bladder diverticular formation, and this all occurs as a result of defect in, and it is an X-linked defect, and is categorized as our and it is an X-linked defect in which the ATP7A gene mutation occurs. So this gene will help in the copper efflux in the intestine. So after the defect of this gene, the copper absorption from the intestine is reduced, and as a result, different enzymes requiring the copper gets defective. So our important reaction occurring in the fibril formation is lysyl oxidase, which requires copper. So as a result, collagen fibril formation is defective and will be share similar features to Ehlers-Danlos syndrome. So in this case, our lysyl oxidase was defective, but here, due to deficient copper, our enzyme is being defective in case of Menkes disease. So Menkes will show similar features of Ehlers-Danlos, but in addition, there will also be low ceruloplasmin levels, and low copper levels. Now let us discuss the elastin synthesis. The first precursor molecule of the elastin will be our tropoelastin, and this structure of tropoelastin is then arranged into a scaffolding protein, which is a microfibrillar scaffolding protein, also known as fibrillin 1. So this fibrillin 1 is like a long rope for attaching our clothes in order to like dry them in the sun, and we now arrange our tropoelastin in these scaffolding proteins, and after that the same enzyme as in the collagen synthesis now acts upon the tropocollagen structure to form cross-linking in the presence with the help of the desmosomes. So this lysyl oxidase will form cross-linking between the lysine residues of our collagen structure in order to form the elastic fibers, our final structure. So let us first talk about the first disease. So if in case of Marfan syndrome, there will be defect in this scaffolding structure, and as a result it will present with defect in the three structures: our heart, our eyes and in the skeleton. So in case of heart, the presentation can be variable. So in case of heart, we can have mitral valve prolapse, aortic dissection, and aortic wall dilatation. In case of eye, we can have cataracts. And in case of extremities, in case of skeleton, we can have long extremities or pectus excavatum, or osteoporosis, arachnodactyly, and scoliosis. So this Marfan syndrome occurs as a result of FBN1 gene mutation, which is autosomal dominant disorder. So the end result of our elastin is an elastic structure which provides elasticity to the soft tissues, and it occurs as a result, in contrast to collagen, it occurs because very few lysine residues are very few lysine and proline residues are hydroxylated, and as a result it does not form triple helices, so it is free to move in contrast to the collagen. So it is highly elastic structure. Now let us discuss about another disease, which is our alpha antitrypsin deficiency. So these elastin structures are often broken down by elastases, which is normally inhibited by alpha-1 antitrypsin. So elastase is often inhibited by alpha-1 antitrypsin, but is now, in case of smoking, this alpha-1 antitrypsin is defective, and as a result elastase activity is unchecked. So this elastase results in unopposed elastase activity will result in destruction of the elastic fibers, and often is presented as emphysema. This is about collagen and elastin.